Assalamualaikum and hi everybody. In this video, I'm going to show you how to analyze open field tests using Smart Software Tracking System. For those who aren't familiar what Smart Software is all about, it is a video tracking system software that allows the researchers to evaluate behaviors in experimental animals. It is easy to use and very user-friendly. So once you open the Smart software, you'll see a few experimental template options. I personally like to choose Customize option simply because it is more flexible to customize our experimental video. You can also use Customize template to analyze almost any type of behavior test that is not listed here. Once you already choose your templates, you will notice the Experiment Assistant button here. Under this button, there are other buttons that will guide you step by step of the whole analysis process. Simply follow each of these steps to do the analysis, and I will explain in detail each one of them later. In order to analyze a behavior test, you'll need a video. Therefore, the very first step is to import the video from the image source. You can either choose a live image by choosing the camera that is linked to the software. Or you can also add a recorded video by selecting digital video file. To add, click on the digital video file button and select the desired video from the folder. Once the video is added, the software will give you a default setting which consists of one arena, one zone definition, and also a digital video control to control the video. The second step is to calibrate the video. To calibrate, click Calibration and make sure the image appeared on the calibration editor is similar to the image of the video that has been opened. If not, get a reference image by clicking on this button. Next, choose the calibration unit that you want to be measured. You will also need to adjust the wanted area by moving the green and red lines. Click Accept once you have done calibrating. The third step is to define the arena. One arena can only be used for one rat. If you need to analyze more than one rat on the same video, you might need to add more arenas. But since I'm gonna analyze only one rat, I don't need to add another arena. Once you are done, click Accept. Step number 4 is to define the zone definition. To edit the zone, click on the Zone Definition button and you'll be directed to the Zone Editor panel. Here, you can draw many different shapes of the arena and zones. You can also choose different color to differentiate between different zones. Optionally, you can also choose a variety of available zone templates. I'm going to select this one. Once the template has been selected, draw the zones by clicking and dragging the cursor over the desired working area. You can further readjust the zone size to fit the working area. You can also manage each of the zones that has been drawn. Open the Management dialog and make sure the zone name, type, and color are correct. Here, you can further label or edit the zones. If you are analyzing other tests such as Morris water maze, you can also label one of the zones as the target. If you are happy with the zone conditions, click Accept. The fifth step is to set the setting for detection. To do that, click Detection Setting button. You can enhance the video lighting by adjusting the brightness and contrast. You may also select Fine Setting to set a special lighting condition that is specific for each zone. Under the Detection mode on the right panel, select both Track and Activity Detection. For track detection, you can either choose Center of Mass, Manual, Tri-Wise, or Color. I'm going to use Center of Mass for this video. Next, under Track Detection, click on the snapshot to obtain the reference image. Before doing this, make sure that there is no rats inside the working area that we are going to measure. After that, click Start Test. While running the test, you need to observe the red tracking line inside the defined zones. Try to get the best detection by adjusting the threshold and erosion scales. Make sure it only detects the red movements. Once you are happy with the detection, stop the test and proceed with the activity detection setting. Click Start Test and try to get the best activity detection by adjusting the threshold scale. Once you are satisfied with everything, click Accept. Step number 6 is to set the time. To set the starting point of the test, play the video until it reaches the time point that you want. Pause the video and click Get from Video button. You will notice that the beginning part of the video has been omitted, and the data acquisition will be started on the time point that has been set from the video. Under the trial acquisition, you can either choose free running, or you can also preset the time if you need to set the time frame. I'm going to choose free running. After that, you need to choose how you want the video to be ended. Under the stop condition, choose either of these three conditions. Once you have done, click Accept. Step 7 is to list down the subjects. Click on the subjects and it will give you all of the details of the experiment subjects. You can import the subject list if you have already prepared beforehand. You can also add, modify, and delete the subjects. Once you are done, click Accept. Step number 8 is to schedule each of the subjects. To do that, click Scheduler. Under Trial Scheduler, you will notice that the first subject has been scheduled for the analysis. To add the next subject, click on the subject that you want to add. Then, click this twin arrow button. As you can see, the second subject has been added and scheduled after the first subject. With all of the settings done, we can now proceed with the data acquisition. Click on Data Acquisition. Under the time control, click Start and wait until the data acquisition reaches the end. If your video is very long, be sure to select as fast as possible option on the video controller to make it faster. Once it is done, you can see the data obtained from the video. Go back to the scheduler and you'll notice that the color of the first subject has changed to red and the second subject has been changed to green color. This means that the data acquisition has been done on the first subject and the second subject is now scheduled for the next data acquisition. If you wish to proceed with the second subject, you'll need to repeat the process by adding a new image source to the software. However, in this video, I'm just gonna straight away do the analysis with only one subject. The last step is the analysis. Click on the analysis button. After that, click and drag the subject that you wish to analyze into the table next to it. Click on this three dots button. Next, click on the green plus button and select Summary Report. Then, click Accept. The next step is to choose the calculation that you want to be included in the report. You don't have to choose all. Instead, choose only what are significant for your test. Once you are satisfied, click Accept and close the report definition box. Next, click the red tick button and then click Analyze. Once it is done, click OK. Click on Export to export the report to the Microsoft Excel. To save trajectory map of each subject, click Export Trajectory Image button and save the image in your folder. Now, let's open the folder to check the files. If everything is there, you are good to go. Well, that's wrap up this tutorial video. I hope this video has been beneficial to each and every one of you. Thank you for your attention and have a blessed day ahead.